So my name is Bristena Ionescu, and I've just completed my PhD in the Department of Clinical Neurosciences. And I'm delighted to talk to you today about some of my work as part of my PhD, which entails modeling progressive multiple sclerosis in a dish using iNSC technology. So a little background about multiple sclerosis. This is a chronic autoimmune-mediated demyelinating and neurodegenerative disease of the central nervous system, which affects about 100,000 individuals in the UK. It affects young individuals because the disease onset is between 20 and 40 years of age. And from a pathological standpoint, the disease entails inflammation, demyelination, and neurodegeneration. It usually manifests initially as a relapsing, remitting disease course where spontaneous repair occurs. However, the disease invariably progressive to a progressive stage, which is characterized by a failure of repair mechanisms in the central nervous system. And this leads to ambulatory problems in about 70 to 80% of patients, which start about 25 years from disease onset. MS is the most common cause of chronic neurological disability of the early to middle adult life and is associated with tremendous costs, both directly and indirectly in the UK, of about 1.5 billion per annum. So as I was telling you, progressive multiple sclerosis initially manifests as a disease that is primarily driven by inflammation. And at this stage of the disease, the brain can heal itself and repair itself. And for these early stages of the disease, we already have therapeutic options. So we have anti-inflammatory therapies, which are able to target the mechanisms at play in the early stages of the disease. However, for the latter stages of the disease, they are largely driven by neurodegeneration, and that namely results from neurons dying. So it's a result of neuronal death. We virtually have no therapy. So there's no current neuroprotective therapies for the progressive stages of multiple sclerosis. So the patient undergo unrelenting accumulation of disability, and this is refractory to current treatments. Aging plays a key role in the progression of the disease, and in fact, it is considered one of the key drivers of disease progression. And recent findings have pointed out the fact that when comparing the chronological age and the biological age of the brain of patients with multiple sclerosis, there is a discrepancy. And this is even more apparent in multiple sclerosis than in other neurological diseases that are canonically thought as neurodegenerative, such as Parkinson's or dementia. The reason why we have a limited understanding over the mechanisms that drive disease progression in MS is because we lack the right models to ask the right questions. This is because MS solely develops in humans. It does not develop in any other animal. And most of the research so far has used animal models, which mostly are able to recapitulate the inflammatory, the initial stages of the disease that are driven by inflammation. They do not recapitulate the neurodegeneration seen in MS, nor do they model aging, which is essential to model when trying to understand what drives a disease that is such intimately linked to aging. To address this issue, recent research has focused more on trying to use human models and also to capture aging. And up until now, it was difficult to capture aging because we were able to design human models by using cells that were coming from patients with MS and using reprogramming technologies to reprogram these cells into an embryonic-like state and then differentiate these cells into cells of the brain that we could then use to model the disease. However, these reprogramming technologies that goes through the embryonic stage erases the aging signatures of the starting material, so of the cells that are coming from the patient. Now, in my study, I used a direct reprogramming technology, which bypasses the embryonic stage and retains the aging phenotype in the cells, which is so crucial for the modeling of this disease. So to recapitulate, the cells I've been using are derived from the skin of cells coming from patients, so this is easily to access. It preserves the genetic background of the patient, which animal models don't. It preserves the epigenetic age, which previously reprogramming technologies did not, and requires minimal manipulation, meaning that this is a more robust and reproducible technology. The technology is also stably expandable, which means that these cells can then be used to ask mechanistic questions and really delve deep into the mechanisms that drive disease progression by using this model in a dish. It also means that these cells are multipotent, and then you can derive cells of the brain which otherwise are inaccessible because the brain is impossible to access, to model. This is a little schematic of what I have done in this project, which is basically taking cells from the skin of healthy and patients with MS and directly reprogramming them into iNSCs. And this reprogramming has led to the emergence of a behavior that was reminiscent of accelerated aging. So these cells in a dish recapitulated this accelerated aging phenotype consisting of increase in senescence markers and also an abnormal metabolism, which I will discuss in a bit. Also, crucially, these cells were able to exert neurotoxic behavior, and they were able to lead to the death of neurons, which we also observe in MS. Now, a mechanism that is at play here, and I will be discussing in this talk, is this fascinating organelle that is called the lipid droplet. And here you can see it in this image in purple. These organelles are storages of lipid, but they also play a crucial role in regulating the senescent features in cells. And this is what I have been investigating in my study. This is a short schematic showing that these iNSCs, through the factors that they're secreting, they can lead to neuronal death. So this model is able to recapitulate these features observed in the PMS brain of inducing neuronal toxicity and neuronal death. And you can see on the lower part of the screen, neurons on the left that have been exposed to medium coming from healthy cells that are thriving. They have their normal branching and network formation, whereas on the right side, you can see that this architecture is completely compromised and the cells are undergoing apoptosis, also known as cell death. As I was mentioning to you, this platform is stably expandable, which means that I could use this technology to then interrogate it in a plethora of manners by employing multiomics profiling. So I could characterize the phenotype, the proteins, the metabolites, the lipids, and then by integrating these data sets through bioinformatics tools, actually identify and propose new targets for treatment, and then validate this and see which features of senescence and of aging that are dysfunctional in these cells are actually driven by the metabolic features identified in these cells. And here is an example of this bioinformatics integration of this multiomics profiling, where I could detect that there's a close link between the lipid droplet synthesis, this organelle that I was talking to you about, and the aberrant secretory phenotype that is leading to neurotoxicity. Finally, because this model allows for drug targeting and genetic targeting, it allows for a really elegant mechanistic interrogation of the mechanisms that are at play. And I was able to interrogate which genes are activated in this process, through which mechanisms these genes are activated, and what sort of regulatory mechanisms are at play here, so that I could then target them and salvage this dysfunctional mechanism. And the drug that I've chosen to target this dysfunctional mechanism is actually a drug that is easily accessible to patients because it's used to reduce cholesterol, actually, in patients that suffer from hypercholesterolemia. And I've chosen here just a snippet of the data from my paper, which shows that by treating these iNSCs with this cholesterol-reducing drug, I was able to salvage this neurotoxic effect. You can see here that the network between the neurons is salvaged, and also this marker of cell death, cleaved caspase 3, is reduced back to normal levels. This is a little video illustrating the mechanism that we propose as a result of this model in a dish. So we propose that actually in the MS brain, part of the failure of the regenerative process is due to the fact that these neural stem cells are creating a hostile niche that could be therapeutically targeted through a metabolic intervention. Finally, just a little bit about the value of the iNSC platform that spans beyond what I've been shown to you today. So the platform can also be used to test out cell transplantation and regenerative therapies by transplanting these cells into mice. They can also be used to derive other cell types from the brain and study their behavior and in a cell-specific manner tease out what's happening in the MS brain. Finally, because this platform is expandable, you can do really elegant studies from a mechanistic perspective and can use gene targeting to then really tease out what is happening mechanistically in the disease process. Thank you for your attention, and I would like to thank my lab and our collaborators and also the funding bodies that have supported this research endeavor. And if you have any questions, I'm happy to take them now.
Thank you. That was a wonderful talk. It's really clear. I'm sort of dying to ask, does it point towards a treatment?
So it does point towards a treatment, but I think what I'm trying to get at right now is the value of the platform as a proof of concept. Because of course, I think that there's no panacea drug in any of the diseases right now that are quite complex and hard to get to the bottom of. I think this is one direction that it proposes, this metabolic intervention, and actually this is also looked into in terms of clinical trials right now. But I think the value of the platform lies in the fact that it provides a tool that previously did not exist to try and understand what's happening in these cells from an intrinsic point of view, so what these cells could be harboring that's dysfunctional in an intrinsic manner.
Thank you. I'm sure we have some questions.
Question.
Yes.
This is John Cook, Bristena. Very nice talk. Thank you. It's something you mentioned right at the very beginning, which you said that MS doesn't occur in any species other than human beings, which was a great surprise to me. Is there any knowledge as to why that is?
We don't really know why this disease does not occur in animals, and part of the reason why we don't know is that we don't really know what triggers it in humans as well. It's an autoimmune disease, but for most autoimmune diseases, we were able to identify what is the protein, let's say, in the body of the human that is targeted by the immune system, whereas in MS, we really do not understand what the immune system is attacking to begin with to even start and answer this question. So we don't know what it attacks in humans. We don't know why it doesn't attack this thing that is elusive in animals. So no, in short.
Okay, thank you.
Question at the back.
Can you say whether it correlates in any way with sleep patterns, since those are important in terms of getting rid of toxins in the brain? And the other thing, I think you mentioned cholesterol, and which way does the cholesterol go to improve things? And the reason I ask that is because at my age, 81, the higher your cholesterol, the longer you're likely to live. So it's not always cholesterol being dangerous. What is the situation in this case?
Yeah, these are two very good questions. For the first one, to be fair, I'm not really aware of data pointing towards sleep patterns influencing the progression of MS, but it's just that it's not my direct field of interest. Maybe I'm more focused on the molecular biology aspects of MS, so I could look into that, but I don't know. I don't have an answer for that. As for the cholesterol, generally it's believed that high cholesterol is bad, and high cholesterol is bad in my model as well. That being said, cholesterol plays a crucial role in a plethora of physiological processes. It's a super important structural molecule. And I don't think you can generalize. I think it's a matter of in which context cholesterol is high, in which cell type it is high, and can we target it in a very specific manner, specifically where it goes wrong, rather than making a generalization, if that makes sense. I hope that answers the question.
Thank you very much. You're saying this is an autoimmune response. Am I right in thinking that there's a general rise in autoimmune disease in this population? And is there therefore also a rise in multiple sclerosis in the population?
So strictly from a database point of view, yes, there is more cases of multiple sclerosis right now than it used to be in the past. But that being said, I think we also need to correct for the fact that now we do have more accurate and sensitive tools to diagnose the disease. And we may be simply capturing more of the patients that previously just remained uncaptured in the pool of patients that we report on. So I think that's difficult to assess since the technology is advancing, the technology that allows us to make a diagnosis and an accurate one with the decades.
Well, I think that is probably the moment to thank Bristena for, it was a really wonderful talk. I mean, it's not just the beautiful work you've done, but the way you contextualized what it is that is so challenging to explore MS, and I think we've all learned a lot from it. So thank you very much.
Thank you.